So we talked about antigenic drift on Friday and just now, and now we have to talk about this antigenic drift. And so what antigenic drift refers to is when there are small changes to the proteins in the virus that allow it to change. And this is sort of one of those things that we know this evolution happens as hosts develop immunity. And if we start out and look at these phylogenies, again, remember I scanned this one, I have a better one in just a second. What you see is this ladder, this stepping stone shape, right? And when you look at where the viruses are, you'd have viruses in 1968, 1969, and then 70 to 74, and so on. And you can see that in the different shapes and shading in the tree. Notice that when you look at, even within 1 year's epidemic, you still have variation. So if you look at the 75 to 79, there's quite a bit of genetic variation in that flu virus, even though you're looking at 1 year sample. So there is still some variation there, especially those triangles really, I think, jump out pretty, and even the clear circles do that as well. All right, so this is showing you the slow change as the flu moves through a season. So we can look at, and we did this, I think, in the past, for viruses, especially viruses that get incorporated into hosts and stay dormant for a long time, like HIV. We also can build in these phylogenies of what's going on in an individual. So if you start with the person on the left, you can see that this person has 4 different strains of virus: red, blue, yellow, and gray, and they have this topology. But he may not pass on that infection to his the next person. In this case, if this is HIV, this would be sexual transmission. But he only passes on some of the viruses that he has. And that can just be random, right? That can just be random chance. She then, over time, might have some mutations that are new in her that show up within, and I made them black and white with a black outline as her new viruses. And of course, they may mutate. The black one would have come from the yellow virus and the clear one from the gray.
Dr. Ed?
Yeah.
Why do flu strains die out?
Our immune systems kill them.
Okay, so like eventually everybody in the world's immune system evolves, or not evolves, but kind of adapts to be able to knock out a flu strain? So
mostly.
Okay.
Sometimes there is, sometimes there's, we sometimes have this response where there are so many people are immune, so many people have this response that the flu can no longer transmit. So
immunity with vaccinations. Once enough
partly independent of vaccination, this will happen too.
But like similar to that, where once enough people are resistant to it, it can't be easily transmitted.
Right, because there aren't enough hosts for it to go, right? Remember, the fitness of a pathogen depends on its ability to move between hosts and survive and replicate in the hosts. So if you eliminate hosts, then you actually, the only strains that will survive are ones that can evade our immune system. Okay, so we do get variation within a host, we've seen that, but we also get the within population variation. So this is combining individual hosts. So all the colors coordinate to an individual, but this is a whole population, right? And so of course, if we want to talk about how a virus is spread, this is an HIV virus phylogeny, and it's showing you that what you can say is that at least you can identify the source of an infection. So female 1 probably transmitted this to male 2 based on the color, right? All of male 2's virus strains are more closely related to the green than anything else. Okay, so we kind of looked at that before. But what you can also do with this is actually, so we've been talking only about the topology so far. We can also start talking about branch lengths. So one of the things in this we've talked about is that the length of the branch represents how many nucleotide changes there are, right? And so in this phylogeny with the frogs, you can see that there are some long branches towards the back of the tree, and then at the front end of the tree, there are shorter branches. And so one of the things we know based on that neutral theory of molecular evolution is that we have the ability to detect mutations that appear in a clock-like fashion. So what that means is, let me see, I think this, hold on. I want to go to the next one. Okay, so what that means is we know that nucleotide substitutions happen at some given rate. And when they're neutral or nearly neutral mutations, they accumulate at some given rate in a lineage. If you can identify neutral mutations and you can identify how quickly they arise, then you can estimate how long ago since you had a common ancestor. So in this diagram, they're showing you a DNA sequence from a common ancestor here at the back. And we know that one substitution happened within 25 million years. And then in 50 million years, you'd expect to see one additional one, right? So you can count up the number of substitutions or mutations and estimate a substitution rate or a mutation rate, okay? And from that, what we can do with viral evolution is look at what we would call a chronogram. So this is the same frog phylogeny I showed you. And it's, instead of looking at it based on just the branch lengths, the number of substitutions, what they've done is they've taken those substitutions and converted them into time. So remember that going from left to right on these phylogenies, the way that this one's depicted, is time. And it's always time in terms of generations and substitutions per site, per generation, or per year. And so what you see here is, instead of having all the tips at different places, we have all the tips at the present, because usually these samples are sampled within the present. And then you can use the branch length, this adjusted branch length with that adjusted substitution rate, to indicate time. So in this case, this is millions of years ago. We usually, for things like species groups of frogs, we can usually use fossil evidence to kind of calibrate how long ago something is, right? And so in this case, they're showing you ranges of error with the purple or blue bars, however you see that on your computer. And they're showing you that this lineage, here, let me annotate where I'm talking about so that we're clear. So if we look at this lineage split here, they're saying that this could be anywhere from about 200 million years ago to about 110 million years, no, 90 million years ago, sorry. Okay, so you've got this range of time. So there's an error estimated based on things like how they calibrate the phylogeny, the type of gene they're using, and other things like that. So it's going to depend a little bit on how the gene has evolved, or genome. Okay, so that's the idea. So when you see a phylogeny that has all the tips shifted like at the same point, and it shows like 0 or something, that's usually something that's been adjusted for time. And one of the things people are really bad about is labeling this axis. So this should really say, right? And people don't necessarily do this. So when I was looking for good examples of this with viral phylogenies, a lot of them just use decimal points with no labels. So I found some that actually will show you this with phylogenies and viruses, but you may kind of get stuck on some of these and we'd have to really decipher what they mean. Okay. Okay, so when we look at, so we looked at this one last time. This is not supposed to be showing you a chronogram. We talked about the origin of a virus, and we talked about how the topology of this, like the overall relationships of the viruses to their hosts, like chimps and baboons and what have you, this is showing us a particular pattern, right? So the pattern here is that every time you see a herpes virus, it's sister to something that's within the primates. Okay, but we can convert these to estimate age. This is one where I couldn't find a good one for all of herpes. I could get it for like alpha 1 or alpha 2, but not for everything. So I switched viruses and wanted to look at, okay. So this particular virus is an enterovirus. It causes meningitis. And so this is a really cool one because they looked at, if you were to play with my image in PowerPoint, there's actually 4 different phylogenies dated by an outbreak. So, so these are 3 different outbreaks shown here. One of them estimates the outbreak in 2004 with errors of 2001 to 2006.9. But you can actually see that it's dated within the 2000s, or at least some of these are in the 2000 block, right? Some of the older ones back here where we know the date of an outbreak, we can actually anchor these trees with the known outbreak, like this sample in the middle panel at the top that says Barty Hill. That one is one that has a known outbreak date. So they sample that patient, and they know the date of it, and they can anchor that and anchor the estimation of mutation rates using a virus that they know. Okay. And then another one, this one is measles. Whether you know this or not, measles is a virus that has a mammal cousin called rinderpest that causes disease in livestock. And so this one is showing you viral outbreaks of rinderpest, RPV, measles virus, MV, and then another related rinderpest-like virus. And what you can see is that down in the bottom, this human outbreak from the 1940s, or not, well, the USA 1950 and Iran 1970 originated back in the 1940s with confidence intervals around it from 1916 to 1954. You guys see that? Okay. So these are ways we can kind of estimate the dates of these things, and it really helps us understand the rates of viral evolution. And what I have on this slide, okay, now I'll skip that one here. So there is a whole field that combines epidemiology, ecology, and evolution and phylogenetics called phyllodynamics. It was a term coined in 2004, and what it's used to do is it takes epidemiological information, phylogenetics, and ecological information about populations. So things like population structure. Does the population tend to have more older folks or younger folks? Who transmits more? How often do people have contact with one another? Things like that. And it uses it to predict outbreaks. So the field of phyllodynamics is kind of cool and new, and I want to show you what's happening using it for SARS-CoV-2 in a second. But this is a nice example of the antigenic drift in influenza H3N2 associated with the dates of outbreaks. All right, so you can actually see the pattern as you move along, and you can take any one node, right? So if you're interested in when this part happened, you could draw a line, a straight line down here to kind of make that estimate, okay? That's how old that clade is, all right? Is that cool? All right, so what's going on now is they're actually doing this in real time right now. And so if you guys, hold on a second, let me get out of here and I'll just share something different with you. What I think you guys still can see is the PowerPoint, right? You can still just see PowerPoint?
Yes.
Okay, cool. So let me share, what's the chat saying? Madison, what was your question about? I didn't see it pop up.
Oh, that was from one of the first few slides.
So I don't know, well, I guess it would be gene flow in the sense that one allele is, if you're talking about it in terms of antigenic shift, yes, it would be genes moving across species, increasing variation. So it'd be like recombination. It's the same as in us. Is that what, is that what it was?
Yeah, I think it was it was the antigenic drift or shift. I can't remember off the top of my head.
It's probably shift because drift isn't gene flow, right? Drift is just mutations across populations.
Yeah.
So if you want to think about this in terms of the four basic evolutionary mechanisms, then antigenic shift would be gene flow from one species to another. Okay?
Oh, yeah.
Okay. So what I want to show you guys is something really cool that, why do I have to do this? Okay. Is this. So on this website, and I left the website in the slides, what you should see now is just my browser. right? You guys see this browser?
Yes, it's
So
really big margin.
it should be a really big phylogeny, and it should also, you should also see a world map.
Mhm.
So if you had looked at this about a month ago, let's say we were in spring break and we were looking at this because we're nerds and that's what I was doing. What you can see is that they're showing you the size of each outbreak. Back in the beginning of March, the size of the outbreak in Australia, there wasn't one. The size of the US, which much smaller, and the width of these connections was much smaller as well. So it's pretty intensely a pandemic right now, right? Like it's kind of really clear. But this website's really cool because what you're seeing is additions of new samples, and you can drag your mouse, you guys can't, but I can, drag your mouse over the phylogeny and see the samples. And so just as we were coming in, we were talking about how this can move into tigers. And so this looks like this sample is the sample from the tiger from the Bronx Zoo, which is kind of bizarre, but also cool, right? So that tells us it's the same virus because it's within a whole clade of human viruses, okay? So it's pretty, I mean, obviously when we're done with this, or if you want to go to this website, you can actually play with all of this. If you sign up for an account, you can look at all sorts of different things on it. If we look at the clock, so when you're looking for a rate of change, you often look at this slope of the line. So each of the dots represents an individual genome, and they're showing you how much change is happening. And so right now it looks like about 25 substitutions per year based on what we're looking at now. And that should be the slope of that line. Okay. You can look at a whole bunch of stuff on here, which a lot of people are doing it. You can do this by country. You can do it by location. You can, so down below the image is also the genome. So that whole genome we looked at in the paper. And if you wanted to just look at the spike protein, you could look at how many mutations have happened in the spike protein in all of this by just dragging. And it's really, there's just this one mutation that's really common in the spike protein. So that's kind of just really cool. Down below where this text is, you can see the phylogenetic context of COVID-19 in with all SARS. So if you do that, this is what it looks like. So SARS-CoV-2 is down here. I don't think they've added a whole bunch to this since the big outbreak in the US, but there's a good representation of the US, Japan, China, Sweden, and Nepal. Right, and then it compares it to the SARS virus from 2004 as well. So that's cool. And the pangolins are in there, some bat sequences. Okay, so you can see, and then of course, if you look at this diversity here, right? If you want to, again, look at the spike protein variation across all of the SARS viruses, you can zoom in and play with this. There's, see, it's, if you just look at the spike protein across all of them, there's a ton more variation across SARS, all the other zoonotic diseases, and then SARS-CoV-2. All right, so that's kind of this is kind of one of those things where this database is changing almost daily. They add tons and tons of stuff every single day. Yeah, so Isaiah, what's your question?
It's not really a question. It's more of just like a kind of a morbid realization.
Yeah.
Like, how can we possibly track a disease so efficiently that we can track like nucleotide level diversity on a global scale, and we still can't convince people that it's a problem? I don't know. That's just,
well, I mean,
kind of
part of that is like the whole field of public health is supposed to be very good at communicating to public. And the mixed messages coming from government versus public health are creating a problem. All right, I had a friend this weekend post a video about citizen journalists going out and videoing hospitals and showing that they're not really having as many patients as the news will tell you. And so there's this unwillingness, there's this human behavior that's just really hard to track, right? I mean, those of us who do science, we like this data, it's comforting. right? It's nice to see that there's not all that much variation in the COVID-2 virus, right? Like, it's just kind of comforting. I mean, it is going to evolve. It will change as soon as we have a vaccine. If we shut it down really quickly, it will. It can go away like other things have, but we can't convince people to get vaccines, you know? So there's that. Okay, so I wanted you guys to see this because it's free to use, it's out there, it's fun to play with. You guys know enough to kind of look at some of this. I'm trying to think if there's anything else that's fun to look at on here. I played with it for a while during break. But, you know, there's a lot. So people, the other cool thing about this is you can actually, if you have any knowledge of doing phylogenetics, you can go in and download all the data from here and build your own trees, build your own phylograms, do your own estimates of things. And it's a free database. But it was really built based on, I'm going to stop this one. It's built based on flu virus, right? Because really we thought the next pandemic would be flu, and it's just not. So
Yeah.
So yeah, that's always comforting. I mean, and things like that where people don't understand the science process. I think that gets at Isaiah's question a little bit. When a scientist is wrong, it carries so much weight, and people don't understand that we iterate, right? We do something, we put it out in the public, and then the public changes what we say. The public scientists say based on other evidence, and people don't understand that constant changing. They have this idea that once right, always right, rather than this constant change where we build on each other's work. And it's very difficult to moderate. Okay, so let me get rid of that. I learned quickly that this software only lets you see like 6 of your windows. So if you have more than that open, the whole world melts for it. Okay, so the last thing about phylogenies that I really want you guys to see is that we can use them to depict geographic origins. So there are people out there who believe that this type of phylogeny that's put in a circle is much easier to read than a square rectangular phylogeny. So bear with me while I try to explain this to you guys. So the root of the tree is in the middle of the diagram where that little stick is. Oops. And then it branches off. So this particular line would be what we had been showing as a vertical line. And then you still get the splits coming off that vertical line. And then then you get a split of species here. So these are your horizontal lines. So you still see clades. Clades are pretty obvious to see because they're coming off of, mostly on this diagram, you can see them coming off of a black line. Okay, it's letting me draw again. So here is the root to this pale green color, right? And this is a geographic region that they've labeled that you guys don't really need to know right now. But you can see that this is a root, and so there's all these green ones with one black line in it. That's one clade. Another clade would be, I'm going to circle it like this. It's really hard to use a mouse to do this. Okay, and then another one was yellow and orange. Huh. Oh boy. That. All right, so once they're a little bit harder to me to look at and figure out what you're trying to say with them, but once you kind of get used to them, you can see patterns in them. And so what they've done is they color-coded their branches based on where they got their samples. And the key thing here is that every color represents a different geographic location. And in black is what we know are samples from the Democratic Republic of Congo, or just the Congo. And so with this, what you see, the pattern that jumps out is that in every instance in this phylogeny, you see a new location that is sister to something from the Congo. So if we look at this again, let's look at this yellow one. So in this clade, you see that all the yellow ones are closely related to a lineage from the Congo. And you do see the same pattern in orange. You see it in red, in pink, in blue, And so from this, we can infer that the source of HIV is from a population in the Congo, right? Because every new infection, new evolutionary lineage is sister to something from the Congo. So this phylogeny is built on reverse transcriptase, which is an enzyme for HIV. And so it's one that you expect to actually have a lower mutation rate because it's necessary for the virus to survive. But this one's another pattern you can get from phylogenies, okay? So yeah, I think that's all I want to talk about today, because you have a test to do and I'm tired. And do you guys have any other questions about the test or anything like that?
In a couple of places you talk about, like, you ask like which of the principles of evolutionary biology.
Mm-hmm.
That's like the 4 things we talked about at the beginning, right?
Or anything actually that we've talked about that's an evolution. Like, so we've talked about today, I would say recombination is an evolutionary principle. Phylogenetics is an evolutionary principle. So really think broadly of evolution concepts that we talked about.
So not just like the whole like fire or like pathogens evolve faster than their hosts.
No, not the evolution of medicine overarching themes. No, evolution itself. So like
Gotcha.
Mutation, selection, drift, all that.
Cool.
All right, more broad. Any other questions about the exam?
And the stuff today that we did with phylogeny, we can include that in the one question. Okay.
Yeah, if it makes more sense to something else that you read, yeah, that's fine. I don't care.
This kind of made like a couple more things like kind of click. So
good. Well, it's part of the reason I wanted to have today is that you could read the questions and then see if there's any questions you had as well. We don't get as much FaceTime, it feels like, just because we're here. We're not here, whatever that means, right? Okay, like you guys, I don't know who isn't here. Who's SpongeBob?
Nicole.
Okay, so Nicole's here. So Derek isn't here and Jaquez isn't here. Okay. All right. So I didn't record the beginning with the antigenic drift shift. So if they have questions and you hear from them, let me know. But again, try to do this on your own. I want to see how you guys are doing on your own. All right. Have an awesome afternoon, you guys.
Thanks, Dr. S. See you later.
See you later.
Thank you.